Welcome to the PubMed Daily Review for April 05 SD. Today we scan 1954 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Researcher, Health, and Morphology Trends. This trend explores how researcher attributes, health outcomes, and physical characteristics influence research and patient care. This cluster covers 469 papers. Representative work includes identifying researcher characteristics driving growth in Japanese university-originated deep tech startups, a machine learning approach, associations between body composition patterns, brain diseases and injuries, and risk of epilepsy, a prospective cohort study, and secular changes in cranial morphology and pattern of sexual dimorphism in modern Japanese. A geometric morphometric analysis using post-mortem computed tomography data. Trend 2 of 5, Space, Chemistry, and 3D imaging. This trend explores how advanced imaging and chemical principles enable novel applications from extraterrestrial origins to material science and medicine. This cluster covers 218 papers. Representative work includes improving 3D reconstruction quality for root phenotyping, assessing the impact of camera calibration and imaging parameters, distribution of extraterrestrial nucleobases, other and heterocycles, and their precursors in a sample from asteroid Bennu, and redox chemistry of early Earth and the origin of life. Trend 3 of 5, Microbiome, Gut Health, and Plant. This trend explores the intricate interplay between microbial communities, host health, and plant physiology. This cluster covers 461 papers. Representative work includes diet outweighs vertical transmission in shaping dung beetle larval gut microbiomes. Short-chain fatty acids activate myenteric neurons and delay colonic motility via free fatty acid receptors 2 and 3 in a mouse, and journey towards piloting Helicobacter pylori screen entry to address health inequities in Aotearoa, New Zealand. Trend 4 of 5, Autoimmunity, Musculoskeletal, and Kidney Health. This trend explores the interconnectedness of immune system dysfunction, skeletal and muscular conditions, and kidney function. This cluster covers 565 papers. Representative work includes parental autoimmune diseases and offspring's allergic disease, a systematic review and meta-analysis, the value of nasal nitric oxide measurement in the diagnosis of primary ciliary dyskinesia, and feet in peril, assessment of feet in patients undergoing hemodialysis. Trend 5 of 5, Cancer, Immunity, and Cell Therapy. These papers explore the intersection of cancer biology, immune system interactions, and novel cell-based therapeutic approaches. This cluster covers 241 papers. Representative work includes clinical and translational advances in regulatory T-cell-based strategies in allogeneic hematopoietic stem cell transplantation. Highlights from ASH 2025, Intrinsic immune properties of carrier MSC impact on the clinical outcome of children with solid tumors receiving oncolytic virotherapy, and protein-arginine methyltransferases in cancer. Mechanisms, functions, and therapeutic opportunities. Up next, our first paper out of 5, Discovery of Covalent Ligands with AlphaFold 3 by Yo Shamir and Nir London from Department of Chemical and Structural Biology, the Weizmann Institute of Science, Rehovot, 761-0001, Israel. Let's break down its key contributions. Covalent inhibitors represent a powerful class of drugs and research tools. However, discovering new ones computationally has been a significant hurdle. While AI models like AlphaFold 3 have demonstrated impressive accuracy in predicting how ligands bind to proteins, their utility in large-scale virtual screening for covalent inhibitors remained unassessed. This study aimed to bridge that gap. The researchers utilized AlphaFold 3's co-folding prediction capabilities to assess its performance in identifying covalent binders. They specifically evaluated its ability to distinguish true covalent inhibitors from similar non-covalent molecules. A prospective virtual screening campaign was then conducted against the Bruton's tyrosine kinase, or BTK, followed by in vitro and cellular assays to validate any discovered compounds. Finally, co-crystallography was employed to confirm the binding mode. The study demonstrated that AlphaFold 3's predictions, along with its confidence metric, were highly effective in ranking true covalent binders, significantly outperforming existing docking tools. This led to the discovery of a novel, chemically distinct covalent small molecule targeting BTK. This compound showed potent inhibition in vitro and in cells, importantly maintaining marked selectivity across the kinome and proteome. This research convincingly shows that AlphaFold 3 can be practically applied to discover novel chemical matter, specifically covalent inhibitors. The successful identification of a selective BTK inhibitor validates this approach. These findings pave the way for accelerating drug discovery efforts, particularly for kinases, which are a highly prolific family of drug targets. Up next, our second paper out of 5. Target-stabilized base editors enable robust high-fidelity RNA editing by Taiyan Liu and Zhonghua Liu from Center for Primate Neuromodulation and Neuroimaging, Institute of Biomedical and Health Engineering, Shenzhen Institutes of Advanced Technology, Chinese Academy of Sciences, Shenzhen, China. Let's break down its key contributions. RNA base editing is a promising technology for correcting genetic errors directly at the RNA level. Imagine being able to fix a mutation without altering the underlying DNA. However, a major hurdle has been the occurrence of unintended edits at other locations in the transcriptome. This lack of precision has limited its application, especially for therapeutic purposes. To tackle the issue of off-target edits, the researchers developed a novel system called RECODE. This system utilizes engineered ADR1 deaminase variants that are tagged with degroans, making their stability dependent on the presence of a guide RNA. The core idea is to ensure the deaminase is only active when bound to its intended target RNA, thereby minimizing unintended edits elsewhere in the genome. The RECODE system proved highly effective. It maintained high precision in editing the intended RNA targets while dramatically reducing unwanted edits across the transcriptome. Furthermore, in vivo studies showed that RECODE could efficiently correct a mutation linked to ALS and introduce a therapeutic mutation in the ANKTL3 gene, leading to positive physiological outcomes. In conclusion, the RECODE system offers a powerful new tool for precise RNA editing, addressing a critical limitation of previous technologies. This work not only provides a more robust method for RNA editing, but also establishes a generalizable principle for enhancing the specificity of RNA-guided molecular tools. Future research will focus on further validating its therapeutic potential in various disease models. Up next, our third paper out of 5. Safety and efficacy of malaria vaccine candidate R21/Matrix-M in African children. A multicentre, double-blind, randomised, Phase 3 trial by Charbel El-Baitar and Moussa Cabaret from University of Oxford. Let's break down its key contributions. Malaria continues to devastate communities, especially in Africa, causing immense suffering and death. While progress has been made, a highly effective vaccine is still a critical need. This study investigates a promising new candidate, R21/Matrix-M, building on earlier positive findings to assess its real-world performance and safety in children. This was a large, multi-center, double-blind, randomized Phase 3 trial conducted across 4 African nations. Over 4,800 children, aged 5 to 36 months, were randomly assigned to receive either the R21/Matrix-M vaccine or a control rabies vaccine. The primary goal was to measure how well the R21 vaccine protected against clinical malaria over a year, with follow-up extending up to 18 months in some settings. The results were highly encouraging. At seasonal transmission sites, the R21/Matrix-M vaccine showed a 75% efficacy against clinical malaria over 12 months, a statistically significant finding. Efficacy was also strong at standard transmission sites, reaching 68%. Importantly, the vaccine was well tolerated, with no treatment-related deaths reported. Younger children in the trial showed even higher efficacy, suggesting a potential benefit for the youngest age group. This study confirms that R21/Matrix-M is a safe and highly effective malaria vaccine for children in Africa. Its high efficacy, coupled with its low cost and recent WHO pre-qualification, positions it to significantly impact the malaria burden. The next steps involve scaling up production and implementation, alongside continued monitoring to ensure sustained protection. Up next, our fourth paper out of 5. Program synergistic photochemotherapy and immune activation of triple-negative breast cancer using anti-PD-L1 antibody-conjugated multifunctional nanocapsules by Jiqiang Lin and Jing Du from Department of Ultrasound, Rinji Hospital, Shanghai Jiao Tong University School of Medicine, Shanghai, 200127, People's Republic of China. Let's break down its key contributions. Triple-negative breast cancer, or TNBC, is a particularly aggressive form of breast cancer. It's known for its high rates of relapse and metastasis, and unfortunately, patients often have a poor outlook. A major challenge is that the tumor environment itself suppresses the immune system, making it hard for the body to fight back. Standard chemotherapy also struggles with effectiveness in these cases. This highlights the urgent need for innovative treatment strategies that can overcome these hurdles. To tackle TNBC, researchers developed a sophisticated nanoplatform. These are tiny, multifunctional nanocapsules, engineered to carry chemotherapy drugs and an immune-modulating agent. They're also coated with an antibody targeting PD-L1, a key protein involved in immune suppression. The nanocapsules are designed to be activated by a near-infrared laser, triggering drug release and photothermal therapy. Crucially, this approach aims to induce immunogenic cell death, making the cancer cells more visible to the immune system, while simultaneously blocking immune checkpoints and using photoacoustic imaging for visualization. The study then tested this system in both cell cultures and mouse models of TNBC. The results from the animal studies were very promising. The nanocapsules, when activated, showed a remarkable ability to inhibit the growth of primary tumors and, importantly, prevent the spread of cancer to other parts of the body. This translated into a significant increase in the survival time for the treated mice. The treatment also successfully induced immunogenic cell death, making the cancer more susceptible to immune attack, and a nanoplatform demonstrated a good safety profile in vivo, with no significant toxic side effects reported. This study presents a highly innovative theranostic nanoplatform with significant potential for treating triple-negative breast cancer. By combining chemotherapy, photothermal therapy, and immune activation in a single, targeted delivery system, it offers a powerful synergistic approach. This strategy not only tackles the tumor directly, but also reprograms the tumor microenvironment to allow the immune system to fight back more effectively. The ability to visualize the tumor using photoacoustic imaging further enhances its precision. This work represents a promising step towards more effective and targeted therapies for TNBC patients. Up next, our fifth paper out of 5. Hemostatic Binab-triggered microgels, BK-TriGS, to address bleeding in neonates by Nushin Zandi and Ashley Brown from Lampe Joint Department of Biomedical Engineering, University of North Carolina, at Chapel Hill/North Carolina State University, Chapel Hill, North Carolina, USA. Let's break down its key contributions. Uncontrolled bleeding is a major concern in neonates, especially during surgery. Their hemostatic system is still developing, making them more prone to bleeding. Current treatments often involve transfusing adult blood products, which can lead to serious side effects because neonatal and adult fibrinogen behave differently. This research introduces a new type of microgel designed to specifically enhance neonatal clotting, aiming for a safer and more effective solution. To investigate this, the researchers developed specialized microgels called BK-TriGS. These microgels are designed to specifically bind to neonatal fibrinogen, enhancing clot formation. They tested these microgels first in lab-based experiments using neonatal and adult blood plasma. Then, they moved to an animal model, specifically a mouse model engineered to mimic neonatal fibrinogen conditions, to assess their effectiveness in controlling bleeding after an injury. The results were very promising. In lab tests, BK-TriGS boosted the density of clots formed from neonatal plasma by over 100%. They also made these clots much more resistant to breakdown. Importantly, when tested in the mouse model, BK-TriGS reduced blood loss by an impressive 50 to 60%. This shows that these targeted microgels can effectively enhance hemostasis in a neonatal context. This study presents a significant advancement in addressing neonatal bleeding. BK-TriGS represent a novel, targeted therapy that could reduce reliance on adult blood products and their associated risks. By specifically enhancing neonatal clotting mechanisms, this approach holds great promise. However, further research, including clinical trials, will be essential to validate these findings and translate this innovative technology into clinical practice for the benefit of vulnerable newborns.